soon as
Let me know when they want to start.
go ahead. It's good time.
Okay. So everybody can hear me, I hope. So welcome everybody. So welcome to the second workshop webinar series organized by Plant Cell Atlas, which is funded by NSF. So my name is Daniel Kierzkowski, and I will be doing the introduction here. I'm from the University of Montreal, and my lab works on morphogenesis in the plants. We are using a lot of life imaging tools to quantify growth in plants. This workshop will be dedicated to the introduction of 2D and 3D segmentations by using MorphoGraphX software. This software was developed by Richard Smith's lab, and one of the co-developers is Anne-Lise Routier, who is one of the speakers here. So this is a series of workshops. The first one was already last year in September, so on the basics of PG, visualization and quantifications. You have a QR code to the recording of this meeting. And we hope to have another one in the future about the use of AI in image analysis. So our two speakers today will be Anne-Lise Routier and Amélie from University of Montreal. So Anne-Lise's lab works on biomechanics of plant morphogenesis. So Anne-Lise is actually a physicist, a little bit biologist at the same time, and a co-developer of the MorphoGraphX. And Amélie is postdoc in my lab, who worked quite a lot on 3D segmentations. So Anne-Lise will introduce you the MorphoGraphX and 2D segmentations, and Amélie will follow with 3D segmentations. So before we start, MorphoGraphX software is well described in the two publications in eLife, one from 2015 and the other one from 2022. If you want to have to download the software, you have to follow this link. So you will find all the versions for different operating systems, etc. There's a lot of resources online already on YouTube, for example, developed by Richard Smith's lab, where you can see different functionalities of MorphoGraphX by following this QR code. The data that will be used to present you the functionalities of MorphoGraphX today you can find them on Zenodo, so stacks, meshes, and segmentations. So feel free to download them here. Because software is relatively complex, what we suggest is that you listen to us, watch what the presenters show you. Ask us questions, so there will be a chat box where you can put your questions, and then get your software and practice later. Feel free to write to us questions later on if you encounter any problems, etc. So after this very quick introduction, I will let Anne-Lise now share her parts and introduce the MorphoGraphX and 3D segmentations. Thank you very much.
Hello, thank you, Daniel, for the introduction. So, as Daniel mentioned, this workshop will be...
Sorry, you are in a presenter mode.
Yes? Oh, sorry.
This is the notes version, yeah.
Sorry.
No worries.
Sorry for that. I will just change the...
Yep, now it's good. Thank you.
Yeah, okay, thank you. So as Daniel mentioned, this workshop will be in two parts. And in this first part, I will really show you how to basically start with MorphoGraphX and do the first type of analysis, which is a little bit more simple in a way, and also which is very specific to MorphoGraphX, which is analyzing things on curved surfaces. So first of all, what is MorphoGraphX? So MorphoGraphX is really a software which was designed specifically for analyzing 3D bioimages. Primarily, it was designed for confocal microscopy, and that's really what most of the applications have been so far. But it's also possible to analyze other type of images, such as block face ACM, MRI, or any other type of 3D images, which gives you a good enough contrast, basically, to segment things. So what is a bit special about MorphoGraphX is that it uses CUDA for parallel computing, which means that it uses the graphics card of the computer to make some processes faster. So it can run without CUDA as well. It's the case, for example, in the Windows version. But to get the full power of MorphoGraphX, you really need to have a Linux machine with a good graphics card, which can run CUDA. Okay, so MorphoGraphX was really developed by scientists, for scientists. I'm lucky enough to be one of the persons who developed some parts of MorphoGraphX, but it's really a very big team effort led by Richard Smith. And so this is a free and open source software. It has been used, it's used currently worldwide in many different labs working on developmental biology, primarily for plant science, but also for animal science. And so far it has been cited over 700 times between the two papers that Daniel mentioned. So MorphoGraphX really should be seen as a modular platform, and it's ever growing. That's why there are already two papers, but there will be more coming in the future. So MorphoGraphX includes several libraries like ITK or CNN for the neural network segmentation. Importantly, it also includes a lot of custom tools for growth and shape analysis, and that's what makes it very special. So, for example, in the version 2 of MorphoGraphX in particular, you can find ways to analyze the shape of the organ and to basically use this organ shape as a custom coordinate to analyze growth, for example. And also, MorphoGraphX supports plugins and add-ons, so it's relatively easy for people who know how to program to make new functions in MorphoGraphX and add to it. So it's based on C++. Okay, so MorphoGraphX is a software for 3D image analysis, and 3D image analysis can be approached in two different ways. So you can first try to do full 3D cellular segmentation. And that's possible in several software, not only MorphoGraphX. And so in this type of segmentation, as we see here an example, which was published, this is not MorphoGraphX, this is another software. So the advantage of this approach is that it contains all of the information you really want to extract from your image, of course. But the downside is that it requires very high quality of images, which makes the imaging itself quite invasive in some cases. And it can make it difficult to follow the same sample over time without stressing it too much. And another downside of this approach is that it's harder to analyze than 2D images. So then what's the alternative to that? Well, a possibility is to do 2D cellular segmentation once you project the 3D image on a flat surface. What do I mean by that? If you take a 3D image, an image stack, like here, a confocal stack, you can, for example, in Fiji, you can make a 2D projection from that. For instance, if you use maximal projection. And so you get this 2D image, and then you can segment the cells from that and do your analysis. So the advantage is that it's easy to analyze. There's a lot of tools for 2D segmentation and 2D analysis. It works with lower image quality than with 3D, so you don't need such an invasive protocol to acquire your images. But a very big downside is that this induces some distortion of shape and of the signal. So what do I mean by that? Well, it's a well-known problem with flat projections. So if you imagine that, well, you have an organ which is shaped more or less like a sphere, well, it's well known that spheres can't be flattened. It's a big problem for making maps of the world, for example. We know that any type of flat projection of the world that you make will induce some distortions. So some countries will appear bigger than they are in reality. And so no matter how you take this problem, you will always end up with some kind of distortion when you try to flatten a sphere. One possibility would be, well, you can try to, kind of peel your sphere and to cut it a little bit like an orange peel. But even like that, it will never be completely flat. And if you try to flatten it, this will induce some distortions. So that's the best we can do, and it's still wrong. Okay, so how can we solve this thing then? Because we have this impossible dilemma where, well, if we flatten our 3D image, we get distortions. And if we try to do everything in 3D, well, it can be too difficult. It can require too high quality of images, or it can be too difficult to analyze later. So that's what makes really MorphoGraphX special, is that so far, as far as I know, this is the only software that proposes an alternative approach in between 2D and 3D. And that's why we call it a bit jokingly the 2.5D approach. So what do I mean by that? Well, in MorphoGraphX, you start with your 3D image stack, and you convert this 3D image into a 3D binary mask. What I mean by that is that in this image, it's a 3D image, and everything which is within the object would be a value above 1, and everything which is outside the object will be 0. So it's really like a 2D binary mask, but in 3D. And so once you have that, you can extract a curved surface from this, and that's this curved surface is represented by a 3D triangular mesh. So a bunch of points connected to each other, forming triangles, and it's really these triangles that form the surface of the organ. And once you have extracted this surface, you can project onto it the cell contours. And the projection, very importantly, is done on the curved surface. Once you have this projection done, you can treat this curved surface as a 2D image. You can run the same kind of processes that you would normally run on a 2D image, like segmentation, for example. But this is really a curved surface which exists in 3D. So it's never really flattened. And so how does it work? Well, this projection in 2.5D basically is a projection which is perpendicular to the surface in every point. So if you look at the cells which are really on the side of the organ here, the projection is done perpendicular to the surface. So you don't induce any distortion on the cells which are on the side of the organ. And what is also interesting is that we can choose the projection depths in MorphoGraphX. So you can choose basically what is the band of the signal, which is here represented in purple. You can choose how shallow or how deep this projection is made in order to get the best contrast between the contours of your cells and the inside of your cells. So to illustrate the difference between a flat projection and a 2.5D projection, here on the left, you see a stack which has been projected in Fiji using maximal projection. And you can see that if we zoom in on a very curved region of the organ, you can see that you have this distortion of the signal that occurs. Well, if you look at the same region on the 2.5D projection in MorphoGraphX, you can see that now you have these very sharp contours with no distortions. Okay, so now I will show you how to get to this point in MorphoGraphX, how to extract the surface and analyze the things on the curved surface. So let's start with opening MorphoGraphX. So you can open MorphoGraphX by just clicking on the icon, and immediately you see that you have two windows that open. You have the main window of MorphoGraphX and you have the terminal. So the terminal, which I'm showing now, is actually a very important window. You should never close it. If you close it, you will immediately shut down MorphoGraphX and you will lose everything that you've been working on. And I see Ivan has some experience with this. So don't close the terminal, okay? The terminal actually has some interesting information in it. So you can see whether CUDA is working or not, whether you're using parallel computing on with another method called OMP. So you can really see what is going on in MorphoGraphX. You can also, when you launch MorphoGraphX, you can see how many processes have been loaded into MorphoGraphX. And if there's a problem with loading the processes, you can see that in this window. Now, the main window is really where you will load your data. And so let's load the first stack. So you can just drag the stack onto the window to load it. And now we loaded data in the first stack of MorphoGraphX. So, okay, let's walk through a little bit through this main window. You can see here that you have on the very top, I don't know if you can see my mouse or not. Can you see it? Okay, great. So on the very top here, you have the main menu. Then on the right, you have three tabs. You have the main, view, and process tabs. In the main tab, you have stack 1 and stack 2. For the moment, we will just focus on stack 1. So when I dragged this first stack into the window, it automatically loaded this 3D image into the main stack 1. You can see that you have a main stack and a work stack. I will explain the difference in a minute. And then below you have all that concerns the mesh, so this surface that you extract from the organ, which is represented with as a surface or as a triangular mesh that you can turn on here. Okay, so now we loaded our first stack, and you can see that it's a little bit difficult to see. So that's a main concern for most of new users in MorphoGraphX is that, well, how do I make my my stack more visible? And the answer to that is that you can play on the opacity of the stack. So here in this stack, we have a lot of background noise. And this background noise, which is actually above the surface, is hiding the actual sample, which is below. So if you play on this opacity dial that you see here on the right, you can see your stack better. Now you can also look at cross sections of your slide, so of your stack. Sorry. So if you go in the view stack, in the view, sorry, the view tab here, you can see that you have these clipping planes here, 3 clipping planes, which are really made to do cross sections. If here in the control key direction, you can decide which clipping plane you want to turn or to move. And so if we click on clip 1 here, this means that by pressing your control key on the computer, you will be able to turn the clipping plane without moving the sample itself. So that's what we are doing now. So if you click enable, you will be able to actually see the cross-section. And now by pressing the control key and using the middle button of your mouse, you can actually go through your sample. Okay, so now we go back to the normal view. So you unclick enable on the clipping plane. And now let's move to loading another stack, so the work stack. So here I've loaded, so you see that you can turn off the main stack in order to see better your work stack. And your work stack is really, it is really, how to say, tied to the main stack. So when you turn one, you turn the other one automatically together. And here in this particular example, I've loaded basically another channel from the same sample. So the second channel is basically showing you some nuclear marker. And so you can see here that it's possible to visualize both channels at the same time. You see that they are shown in two different colors. So you can process your stacks, you can save your stacks after processing, which I recommend very strongly because there is no undo button in MorphoGraphX. Remember that, no undo button. So whatever you do, it's irreversible, okay? But what is important is that is to remember to save your the results of your processes as you go. Every important milestone, I would say, requires to be saved. And you can do that in the top menu here. If you look at stack 1, mesh 1, stack 2, mesh 2, you can save independently every stack. So you can save the main stack, the work stack, boosting stack 1 and stack 2, and then your meshes as well, you can save them. Now, another thing to understand MorphoGraphX is that you can also save the session in itself. So that's what I've done here when I clicked on File, Save on the right left corner. Really what I'm doing is that I'm saving the session in itself. And that is different from saving the data. So this is not going to save my stacks. This is just going to save the MorphoGraphX session. And what I mean by that is that now if I shut down MorphoGraphX, so I shut down the main window, I shut down the terminal, and now I'm double clicking on this new file that I just saved, you see that now MorphoGraphX is loading the session exactly as I saved it. So with the same kind of rotation of my stacks, with the two stacks loaded with the proper opacity, etc. So when you save your session, actually it also saves all the parameters that you've been using in the processes. And that's a very important thing to do because when you want to run things in batch, you want to reuse the same parameters, and so you want to be able to find them quickly. And so saving your session is really a good idea. Okay, so now that we see how to use MorphoGraphX, we can start actually processing a stack. So here I've loaded the same stack as before, which is the sample marked with PI, and this will be the T0 time point. Okay, so we will try to extract the surface from this. So the first thing I'm doing before trying to extract the surface is to get rid of the noise. And to do that, I will run the process median of medians. So this is just a median filter. You could use a Gaussian filter instead. I tend to prefer the median filter because it gets rid of the noise while preserving the contours of the sample. And you can see that immediately that when I clicked on this filter, we switched from the main stack to the work stack. That's why it changed the color. So in MorphoGraphX, if you have the main stack which is active, which you can see, maybe I will go back to a little bit in time. So here you can see that at the bottom right corner of this window, you can see that the main stack is active. which meant that in my main window, I had the main stack checked on, but not the work stack. And now if I apply the process that I chose, so here the medians of medians, you can see that immediately after, you have the work stack that became active. That's because the MorphoGraphX applied the filter to the main stack and then it put the result in the work stack, and this work stack became active. Now what you can do is that you can use the process called copy work to main in order to take the result of your operation and put it back into the main stack. That's very useful if you want to take your process stack and then do some tests on it. Okay, so that's what we will do now. We copied our filtered stack into the main stack, and we can do further processing on it. So remember that now our stack has been filtered, so we can go on and try to extract the surface from it. For this, we will use the edge detect process. This edge detect process really turns the stack into a binary image. Now, the way this process works is basically it goes from the top of the image towards the bottom, and it detects the surface as it goes. Which means basically that if you have a surface which is really a little bit curved on inwards on the side, this process will create blocky edges. Yes, it will not be able to extract the surface that goes a little bit inward. So to solve this issue, we can use another process which is linked to edge detect, and which I will show you in a moment, which is called, oh, sorry, yes. So before moving to that, I changed the threshold in the edge detect in order to go a little bit deeper within my sample. So you have to play on this threshold to make sure that you extract exactly what you want and that you don't get too much noise. And so now let's look again at the result of the edge detect. So you can use the keeping planes to look at the result of the edge detect. And you can see that after increasing the threshold, the image is cleaner, but we still get the edge of our sample, which is not curved inward as it is in the actual data. So we want to be able to curve a little bit our 3D binary mask in order to reflect better the actual shape of the sample. And that's what we will do with the edge detect angles that I'm using now. So when you use the edge detect angle, you should have the main and the work stack both clicked on. And then you can turn the stack. So you should have the stack one active for the control button. And if you just turn the stack one, now you will be able to basically to take out a little bit of some chunks of this binary mask in order to fit better to the actual shape of your sample. So let's see now how this looks like from the side. So now you can see that on the left here, that our binary mask is now fitting better the actual shape of the sample. So it's a little bit going inwards. Okay, so now we have our binary mask, and we are able to extract the actual surface from this, yes, because so far we have been working only with 3D images. So now we want to make our triangular mesh to represent the surface. So we move into the mesh process tab, and we go into the creation menu, and we do the marching cube surface. So here with the marching cube surface, I use the cube size of 5. And that means really that the mesh will be quite coarse. Before processing the mesh further, I want to reduce its size a little bit. So I want to trim things which are not necessary. Like for example, here the bottom of the mesh and the sides are not needed. And so I can just trim them away to make the mesh a little bit lighter. Okay, so now I continue to trim the mesh. I can also trim the bits which are on the side and are a bit ugly. And so once I've done that, it's always a good idea to use the clipping planes again and check how far is my mesh from my actual sample. And you can see it's a little bit up from the surface, but that's okay. As long as the distance between the mesh and the sample surface is consistent, you can consider it's fine. So, okay, so now I've done some smoothing of the mesh, and smoothing is really important right after you extracted the mesh, because that will create a nice even spacing in between the points of the mesh. And you can see here that, so if we zoom in, actually, you can compare the size of the scale bar here with the size of the triangle. So you can see that I used a marching cube size of 5 microns, and this gives me really triangles which are about twice, half this size, so 2.5 microns. So now we can dezoom, and what we want to do is to check that we don't have any things hanging from the mesh. Okay, so now the next step is I want to project my signal onto the mesh. And here the mesh is still very coarse at this point. So that's why you don't get a very nice image. Now, if I select all the mesh, you can see here that when I selected an entire mesh with this tool here, you see on the bottom left corner the number of vertices. So I have 13,000 vertices, which means 13,000 points forming my mesh. So this is a fairly small mesh at the moment. So it's very coarse. It doesn't have a good resolution yet. So next, what we want to do is really to refine the mesh progressively. So to refine the mesh, we will use the subdivide function. And you can see after using the subdivide and smoothing the mesh to evenly space the vertices, you can see that now the mesh is much finer. And so when we reproject the signal, we get a much better resolution on our surface. Now we can check again the size of our mesh, and you can see now that we have 52,000 vertices. So we already multiplied several times the size of the mesh, but it's still a fairly small mesh that can be easily manipulated within MorphoGraphX. So you could subdivide multiple times your mesh until you get a really, really fine resolution. The problem with that is that the bigger the mesh, the more points you have in your mesh, the harder it will be to process it in MorphoGraphX, the longer it will take. And so the the whole trick is really to find exactly the right resolution that you need while avoiding to get a too big mesh. So here we are at 210,000 vertices. This is still manageable, okay, but you don't want to go much larger than that unless you have a really, really powerful computer. Okay, so now that we have our signal which has been projected on the surface, so you can notice here the projection. So we'll go a little bit. Before that, yes. So here you can notice that in this I use the signal projection process here, and you can actually change the distance parameters that you have here. So what do this parameter mean? The minimal distance that you see here is 2 micron. That means that, and the maximal distance is 6. That means that for every vertex in my mesh, I will basically take all the signal which is situated between 2 and 6 microns away from this vertex and project it onto the surface. Again, the projection is perpendicular to the surface. So it's really taking a kind of column of signal situated between 2 and 6 microns away from the surface and projecting this column of fixed signal on the surface perpendicularly to the surface. And so it's really playing on these parameters that you can get a nice signal, a nice contrast on your mesh, which is really needed for the steps of segmentation. So now, okay, sorry, it's going a bit too fast. So, okay, so before we segment, we need to blur a little bit the image. And that's because when you blur the image on your mesh, so which is done, this blurring is done with this here signal smooth machine signal process. So by blurring it, we will be able to better detect the cells and then do the segmentation. So we blur it a little bit, and you can see here that I used 3 passes of blurring. Okay, so now our contours are a little bit blurred, and now I'm using auto-seeding for the segmentation. So what auto-seeding does is that it will basically put some seeds of labels. So it will basically assign some random numbers to to specific points on the mesh, which you see here as colors. And the seeds are placed in places which are darker than the other places on the mesh, which means that here these seeds will be placed mostly in the center of the cells. That's why it's important to blur a little bit the signal before you do the auto-seeding, because when you blur it, it becomes easier for the program to find the center of the cells. And so by doing this auto-seeding now, we have mostly every cell which has a single label, but sometimes we have cells which have multiple labels inside them. And we will see how to deal with this issue later. So once you do this auto-seeding, you can run the watershed segmentation, which will propagate the labels in order to fill the entire cells. And that's how you can get your cells. Now, in order to deal with over-segmentation, oh, sorry. The problem is speaking. Okay. Okay, so in order to deal with over-segmentation, you can use the combine labels, and that will basically fuse together cells which were over-segmented. Now we can look at the contours of the cells here by clicking on mesh and cells. That will show you only the part of the mesh which are really defining the borders between the cells. And you can see that some of the cells were under-segmented, so which means that they were fused together. So we want to correct them. So here what I'm doing is that I'm just filling the cells which were under-segmented with an empty label. You see here the bucket, and here the color was empty. Now I'm using this tool to put some new seeds for a new segmentation. And I can run again the watershed segmentation, and now I get a properly segmented mesh. And I can continue to correct my mesh. Okay. I will skip a little bit this part, but if you want to refine your mesh without having a too big mesh, you can also use this function, which is subdivide adaptative near borders, which basically means that if you have already cells which are segmented, if you run this function, now you can refine the mesh only in the places where the cells are touching each other, so on the borders, which allows you to get a mesh which is coarse enough in the middle of the cells so that you don't get a very big mesh, too many points. And then you have something which is refined only on the contour. Okay, so let's move on to the next step. So now we have, so we already have our segmented mesh for one time point. So what can we do with that? So once you have your cells which are segmented, maybe you want to analyze their shape. And so you can do that here in the mesh cell axis and then shape analysis process. And if I run that, basically this runs a PCA on the cell shapes. So again, this is 2D, yeah, so I will get just 2 axes. And you see here in the display shape axis that you can basically change the way you display this analysis. So the analysis is still the same, but you can choose to display different things as a heat map. Maybe you want to display this, only the main axis, etc. Now, what you can do as well, if you have only one time point, is that you can choose to basically look at signal quantification. So let's load here our second stack. In the work stack, we load our second channel, which was a nuclear marker. And once you have your nuclear marker, you can actually project it as well onto the surface. And here I choose to project it between 0 and 10 microns in order to get the signal from the entire epidermis. So now I have my cell contours and I have my projected nuclear marker. And so I can really analyze the strengths of this marker for each individual cell. So I can analyze the total signal. I choose signal average here to make sure to normalize the signal by the area of each cell, basically. And now I can get this this quantification of the cellular signal. Okay. So another strength of MorphoGraphX is that all of these things that you can visualize as heat maps, you can save them as CSV files. So in other words, like you can make a kind of Excel table out of it, where in one column you will have the labels of the cells, in another column you will have whatever value you want to quantify. And then you can compare things with each other. So let's say, for example, you want to compare the cell shape with the signal strengths, as we saw here, you can combine these two things together in Excel, for example, and then you can load the result of your computations that you did in Excel back into MorphoGraphX. So I will try to go quickly on how to do lineage tracking in MorphoGraphX. So, okay, let's say that you have your first tag that you already segmented. Now you can also load something in stack 2. So stack 2 is basically working the same way as stack 1, and it's just a separate kind of space for you to store a main stack, a work stack, and a mesh. So let's say that for our second time point, we already segmented the cells. And so you can see that here we have these two segmentations which are independent from each other. So if we were to compare the values of the labels on one mesh and on the other mesh, well, even if we can identify the same cells, we can see that these labels are not the same. So here with the pick tool, I'm trying to look at the values, and I can see that, well, the value here that I have for this cell on this mesh on the right is not the same value that I have for the other mesh on the left. So which means that, okay, we have these two independent labeling. So how can we make the connection between them? Well, for that, we will use what we call parent labeling. So if I click here on cell label parents, and I can actually get into another type of labeling. And this is what I will use to make the connection between the two meshes. So very quickly here, I'm changing the for the mesh one on the left. I just visualized the mesh itself and not the surface, and that allows me to basically align the two things together in order to identify the same cells. So you can see that I'm turning the meshes around in order to look at, okay, how the cells in mesh 1 correspond to the cells in mesh 2. And it requires a little bit of practice. And now I see, oh, okay, well, the cells are well aligned with each other. So now we'll use this tool here, which is the grabbing tool, to basically grab the label from the first mesh and apply it to the parent label of the second mesh. And so once you do that, basically, this is a way for you to show that there is a correspondence between the two meshes. And you can save. So, okay, so let's keep that a little bit. So once we have done this parent labeling, you can notice now that the cells have similar colors between the two, yes. But so on mesh 1, we have the actual labels. On mesh 2, we have the parent labels. And you can see how they correspond to each other. And actually, we can save. So these parent labels are saved with the mesh, but you can also save them separately as a CSV file using this save parent process. So now saving the parents. And I can also load the parents for my next analysis. So, okay, now let's say you have your first mesh segmented, you have your second mesh on which you have already done some parent labeling. And here you can see the complete parent labeling. And actually, in order to check if your parenting is well done, what you can do is to create a heat map of growth. So maybe I will skip to this because we are running out of time. Yes, so if you do a heat map of growth between your two time points, you can notice that at the first run, probably you will get something which looks like this, where you have some obvious outliers. And so these outliers actually correspond to errors in the parenting. So what I would recommend to you is to basically you identify your outliers on both meshes and you use this select by label tool in order to select the contours of the cells. And that will make it easier for you to correct the parent labeling. Okay, so I will maybe skip that. I'm very sorry. But yes, so here we correct the parent labeling by again putting together the meshes one on the other, and thanks to the selected cells, we can see immediately where to do the correction. And so once we have corrected all our parents, we can actually compute the growth, which I will just show you here. So you go again with the heat map computation. And you can see now that you have these beautiful heat maps that look exactly the same on both time points. And that's what you want to make sure that you don't have any outliers in your parenting. So once you have done that, you can also go into more advanced functions like computing the directions of growth. Unfortunately, there is no time for demonstrating this, but don't hesitate to go into the MorphoGraphX help section. There is a tutorial that will show you how to do all this more advanced analysis. So thank you. Sorry for running over time a little bit. And yes, and now Amélie will show you how to do the 3D segmentation.
Okay, so I'm going to share my presentation. Do you see my screen? Yeah. Okay, so thank you, Anne-Lise, for this nice introduction into MorphoGraphX and also to this 2.5D analysis. So welcome everyone to the second part of this workshop that is going to be more focused on 3D analysis on MorphoGraphX. Okay. So what we will discuss together today, so why 3D analysis, why it's useful. I'm not going to go really far into why we are using MorphoGraphX, as Anne-Lise already discussed a lot about it. What are the thing that we need to know before doing image acquisition, and what you have to think before that to do 3D image analysis. Then I will discuss a bit about the thing that you need to know before starting to do the proper analysis, and then I will do the live demonstration. Oh, sorry, I have to move that. So why 3D analysis? So first of all, because it's so fun and you have a lot of time to spend in 3D analysis, but also because it's really important to do volumetric measurement. And with 3D, you will be able to study cell shape, such as cell sphericity and isotropy, or to study the elongation of cells, but also to do cell volume analysis. It's also important to follow the behavior of cells, such as the evolution of cell shape, the cell division, and also to study differential growth between cells or tissue or even cell layers. So I will not go into big details here. I will just remind you that it's a software built for plant biology. It's very useful for 2D and 3D image analysis. You can do some time-lapse analysis in this software, and I will just remind you that it's an open source and really well-maintained software. So what you need to know before doing your confocal images. So here is an example of the images that you can have. So here is an Arabidopsis gynosium with a PM marker here. So first of all, you need a really good fluorescent signal. So that's your first point. You really need to have, for example, a nice PM marker, a fluorescent marker, or a strong PI to stain the cell wall. Otherwise, you will have difficulties to do the segmentation, or probably you will fail because it's too long, too complex, or you will just destroy your mind. So be careful about it. That's the really, really important point. You also need to acquire your images in 60 bytes. That is something really important to keep in mind. The second thing that is really important is to think about the voxel size. So here we are doing 3D analysis. So you need to do a Z-stack images in deep in your tissue. So you will not stay in 2D, but in 3D. And the voxel is basically a pixel in 3 dimensions. So you are adding the Z dimension, and you will need to have this voxel as close as possible to a cubic shape. So I know that there is some limitation when you are doing image acquisition, and also it depends on the size of your sample. You cannot spend days on your confocal, so sometimes you cannot have a perfect cube voxel. So try to have about 70% of the voxel size that is representative to a cubic size. And then if you are doing any time-lapse experiments, so here I just wrote that it is optional. Just make sure that you repeat the experiment on the same sample, same region, same cells, and continue your experiment on that. So then you will need, of course, MorphoGraphX that is working better on Linux and Microsoft. Sorry for you, but it's not working on Mac. You will need to pre-process your images on Fiji. And for the 3D analysis, you will need the ITK module that is necessary for the 3D segmentation. So before doing anything, we make sure that we have near isotropic voxel, 60 bits image, convert your image in TIFF. And then here is just good to know that there is an active ROM and another kind of MorphoGraphX that is working. So let's talk about the demonstration today. So here I'm going to focus only on the first cell layer, and I will present how to do cell segmentation and how to correct it, but also to create this 3D cell mesh that corresponds to the surface of each cell and not only the surface of the organ. Then I will show you how to do the cell volume analysis, and if we have time, we can also discuss about how to analyze a time-lapse experiment in the first cell layer, but also how to extract the information from the second one. So here I will present you a confocal image of an Arabidopsis thaliana plant, and more specifically an image from the gynosium organ, and I will focus on the shoulder of this organ and in this area. So here is the image I'm going to use today. So let's start the presentation. So the first step is definitely to open your image. So as I said, you need to convert it in TIFF before opening it in MorphoGraphX. So make sure it's already the case. Then you can visualize your sample. Here is inverted, so I just changed the orientation of this sample. As Anne-Lise already described, the first step that we have to correct or change is what we definitely see. So there is two ways to change the intensity of the signal, is to modify the histogram here, or to modify the opacity of the sample. So here is just a way to show you how to change a bit these parameters. And then before starting the 3D segmentation, we need to blur the signal that will allow the detection of the plasma membrane marker. So here I'm using the Gaussian blur, so it's a good example to show you that there is different way to use filters. So Anne-Lise used the median one, and here I'm using the Gaussian blur. I'm not changing any parameters here, and for now it worked pretty well. So as she explained, there is multiple space of work. So the main will be your original image. The work that has been created in blue here is what we call the work. So it's your work environment. It's where you are working basically in the software. And each time I am doing something, I am saving the images, because you can lose your analysis, or you can have morphographic crash that happen. So make sure that you save at every time point your analysis. So here to save the work, you just click on stack one, because here I'm working on stack one in the work section, and then save. So here I just rename it with the work name. Yeah, and then to do the 3D analysis, I'm going into the process, the ITK section. As I said before, it's the ITK module, and then segmentation here. Here there is the ITK watershed auto-seeded is more or less the same thing as in 2D. So it's going to be automatic, and it's going to place one seed in one cell in the middle of the cell, and then extend the label to the whole cell. So here the value of this ITK is really important, but I'm not changing the value for this sample, but you will have to change it and find the good one for your marker, for your sample. And I will show you how it is affecting the ITK segmentation later on. So here I'm just running the segmentation. It's taking some time. And so I will just, yeah. So then don't be surprised that you will have this kind of weird thing. So here it's a cubic segmentation. It is also detecting the outlines of the image, and definitely we don't care about this part and this part, so we'll remove them. So how to remove the information we don't need in the segmentation is to, it was to use this tool and click directly on the color, so the label that we don't need. So here is the results of our 3D segmentation. And what is really important here, and what I wanted to show you, is that I will play a lot with the different parameters in the main and also the work to correct this segmentation. So as I said, the value of the ITK that I used is really nice for this sample, because it's not over segmenting the cell, but not also under segmenting that much. We can see that we have a really nice segmentation in these cells, for example, but we still have some issues with over segmentation, which means that we have different label in one cell, and we know that it's not true because we clearly see the boundary of this cell around here. So we definitely have to correct all this to have a proper 3D segmentation. So here is a nice representation of the ITK segmentation. I'm just showing how to change the opacity to make sure that our observation is true. I pick up the label, for example, of the orange color here to give the same label to the rest of the cell. So as you can see, we are clicking on a lot of buttons in the software, but also on our keyboard and on the mouse that we are using. At the end of this presentation, there is a screen with all the keyboard piece that we are using, because I know that sometimes it can be confusing to know which one to use and how to use the shortcut and everything. So here is just a correction of the segmentation, then zooming, make sure that everything is well segmented. So it's a lot of eye analysis. You have to take time to be sure that your sample is well segmented. Yeah, so then the next step, oh, okay, how I can move that, will be to move in your sample, because here you can see that all the cells are well segmented. I'm not really interested in these cells that are at the boundary, and you will see that I will remove them later on. But for the rest, we can see that all the labels seems to be good, but we are just looking at the surface of these cells. So here you definitely need to go into your tissue and see in an orthogonal view how the segmentation is. So here I will just show you how to move into this direction, but of course you will have to do that also in this direction, but in Z as well, to make sure that you don't have any segmentation errors. So to move into your sample, you can use the clipping plane as Anne-Lise shown before. So to use the clipping plane, you have to click on View, Grid to make sure that the grid here is appearing. And once you click on Enabled, it's doing the cut into the sample. So I'm just placing this grid into the sample and try to move along the axis. So here I said before that I'm more interested in the first cell layer, so I'm not going to show you how to create the second cell layer, but basically it's the same. So you can directly correct here, or only the first cell layer. It depends on your needs. So here I'm just traveling along the sample and try to find errors in the segmentation. So here is a good representation of what can happen, because every label is represented by one color, but here you cannot really know if it is just one cell and if it is an error of segmentation, or if it is 1, 2, or 3 cells. So to check that, I just removed this by using this tool. I removed the label of the cell and check that it's a real cell. So here also it's a good example to show why it's important to save your data. So before moving into the same sample, I saved this ITK in the work also, to make sure that I can come back to this ITK in case I'm doing a mistake here. So then I just load again the ITK. And I continue to move into this sample to correct. So I didn't find any strong errors in this orthogonal view, so I just wanted to correct this one and make sure that this part that is more a plasma membrane marker is not integrated into the volumetric information of the cell. So I removed the label of this cell to create another one. So it's the same thing as into 0.5D. So we are going to use this magic tool. Then go to view, make sure that here fill and seed are clicked. So seed means that you will place a seed here, and fill will fill in the cell in 3D. So here I'm also forcing the software, and I wanted to show you that because I didn't want it to show a perfect image, and we have a lot of wilting in samples. So I wanted to show you also how to force the segmentation to remove this part that is not a real cell volume here. So you can just place a point by modifying the size of the magic tool that you are using, and then it's going to do the segmentation. I also wanted to show that there is 3 kinds of watershed segmentation, and here in this case, the ITK watershed resegment does not work, and you will have to use the ITK watershed segmentation. And here in this video, I think I stopped before, yeah. So trust me, it's going to fill this cell in a proper way. So I wanted also to show you the impact of the value of the ITK here. So if you are using a low value of the ITK, it's going to over segment your sample. And it looks complex at the first time when you see that, but it's sometimes better to have over segmentation than under segmentation, as you can see here, with a higher value of the ITK, because sometimes you can miss some cells or have other kind of problems. So personally, I will prefer to have over segmentation, but of course I will try to have a value between this and this situation to do not have missing cells, but to do not have too much manual correction to do. So then let's talk about the creation of the mesh. So as in 2.5D, the mesh is a triangular surface, and what is really different here is that this mesh is going to be glued to this ITK 3D structure. So it's going to give you the information of each cell volume. So how to do that? So here you click on process, you go to Marshik Cube in 3D this time. I'm changing the value of this cube size to 1. So here, if you use a high value, it's going to increase the size of your file, and no, it's going to decrease the size of your files, and the inverse, if it is lower, is going to increase the size of your file. But here, the value 1 in our work was the perfect value to use. So then it's taking a few seconds to run, and you can see that the mesh has been created here. So first, we talked about this main area that is your original image. We were working on the work when we did the Gaussian blur, but also the ITK volume. And now we created a new space that is the surface that is also represented by the mesh here. So you can also visualize the mesh by clicking here. So I just remove some information because we don't need any more original signal, but we don't need also the work. And I want to see how it looks like in 3D. So in the work, I had the ITK for every cell in different cell layers, but here I told you that I'm more interested on the first cell layer. So how to extract only this cell layer. I will show you that later, but it's also good to know that you can work on different ones. So how the mesh looks like. So to visualize it, you just have to click on the mesh, change the information here, and visualize the whole structure of the mesh. You can also decide to visualize only the cell. So I click on all, it's this triangular surface. But here, as you can see, we are not only working on the surface of the sample, but we have the information of each cell. So here I just click on individual cell to show you that they are well segmented, well separated, and we will be able to analyze the information of individual cells or multiple in one cell layer. So to separate this cell layer from the other ones, I select all these cells manually by clicking on the selecting tool. It's a little bit long. Just accelerate the process. And what I do at this step that is, in my point of view, really important is to save the mesh with this selected cell. Because if you want to come back later and study the second cell layer, you already have the selection of the first cell layer and you don't have to do it again. Because when you save this mesh at this step, the selection is also saved. So if you want to come back, you can just open the file and suppress the first cell layer. So here I save the mesh with the name of our sample. So here it was the time point 7. And to remove the other cell layers, I just click on so Shift, and so I wrote it's Shift key and I on the keyboard, and click on suppress. So then it's going to suppress all the cells, and you will keep only the first cell layer, as you can see here. So again, I save this first cell layer and naming just T7L1. Then it's really important to check again your segmentation, because sometimes, because you have similar colors, for example, between these cells, you don't really visualize that it is a different label. But when you create your mesh, you will see that it's, for example, a mistake or not. So you will have to do again the clipping plane and move through your sample and check your segmentation. So I'm using the clipping plane the same way as before. Here, just to help me to visualize, I'm changing also this parameter to visualize the surface, so the mesh surface here, because it's clearly easier by eyes to decipher the volume of the cells when it's yellow than just colored. So then I move through the sample and I'm trying to find mistake in that. So what kind of mistake you can have is over segmentation or under segmentation. So here I was searching, and yeah. So for example, here there is, here there is a problem, yeah. So here there is an over segmentation. That means that there is 2 cells in one. So I'm just clicking on the workspace to visualize if it was a problem from the ITK, and obviously it is. So here we have 2 different labels represented by 2 different colors. So when we created the mesh, it created the mesh on this cell, and because we inverted the selection and suppressed the other ones, this one disappeared. So here how to correct that? You have two different ways. You can suppress the two colors and come back by placing a seed again in this cell and do the watershed segmentation. Oh, what happened? No. Yeah, the watershed segmentation here. So when you do the watershed segmentation, so it's going to take some time creating again this label outside of the sample that you don't need. You can just erase them. I just save. Oh no, I just wanted to show you the different ways. So the first one was to create a new seed, and the second one is to use the label. So you can use this to give the same label of this cell to this cell, or the inverse, as you prefer. So then I save this new, oh, sorry, I don't know why. This new ITK. Yeah. This new ITK to be sure that we are, we have this one for future analysis. And then I remove in the mesh this cell that we don't need anymore because it was not the proper one. So what happened here? So I removed this cell because they were a mistake. I saved the file. Then I went back to mesh Marching Cube 3D. I did the creation of the mesh again of all cell. But I want to only correct this cell. I don't want to touch to the other ones. So what I will do is to select this one, invert the selection, erase the other ones because I don't need them. And then I will save only this cell into another file and open it into a stack 2. As you can see, I just check that this cell is really gluing our signal. I'm saving it. Yeah. And then I make sure that I will have in the stack 1 the mesh we want to correct with the cell that is missing, and in the stack 2, the new cell that we created with the correction. So then I load into the mesh 2, that is into our stack 2, this alone cell that I called cell. So here we have both of them, the 3D mesh that we want to correct and this cell that we want to be fused to this mesh. So how to do that? You go to process, then it's multi-mesh and add another mesh. And by clicking on that, you are adding this alone cell from the stack 2 into the stack 1. So then you did the correction of your 3D mesh. And of course, you save this correction. So you can have something else that is the under segmentation, and it's going to be exactly the same process. But here, before doing any changes and modification, just make sure that you have a real problem by playing with these different parameters, changing the opacity of your work, increasing, for example, the opacity of your main, and make sure that you have a plasma membrane or a cell wall signal here. So I remove the label of this cell, and then I am using again the magic tool to place the seed inside this one and another seed into this one. and run the watershed segmentation again by clicking on process, stack, and ITK watershed segmentation. And then I don't need the information that is going to appear in this area. So by clicking on that, make sure there is no label, I erase these informations. And then you can see that I have this new ITK structure, but we don't have any changes in the mesh. So to have this change, we have to create again another mesh. Clicking on mesh, Marching Cube 3D, so I can remove this cell. And yeah, run again. And you will see that it's going to segment properly these two different cells. As you can see here. So how to use this information? So then you did the 3D segmentation of each cell. So how to extract the information of cell volume, for example. So first of all, I just removed the cells that has been cut due to image acquisition. So the cells that are at the border here that I don't need, by just selecting them and suppressing them. I save the folder again as my final image. And then by clicking on, so sometimes you can have weird shape on your mesh. And here was a small example of things that you can avoid in your mesh. So there is a kind of triangular shape here that does not really look like biological. So that was a good example to show you how to create also weird things. But please make sure that it's also fitting with your PM marker or cell wall. But here it was just for the demonstration. You can select this tiny zone, then go to process and play with the structure. You can then smooth this structure to remove this kind of triangular shape. So then to have the information of cell volume, you can go to process, heatmap. We are not going to extract the information from this dialog window, but in this one, that is the heatmap classic, where you can extract the volume information here. So then click on OK, and it's going to give you a heatmap of the cell volume, as you can see here, with a smaller cell volume in this area, for example, and bigger cell volume here represented by the color scale here. You can extract this information by saving this into a CSV. So on heatmap, then heatmap save. And you can give the name of the information. I don't know if I still have time to do the lineage tracking. What do you think, Ivan? Do I have time to?
We're now at hour and a half designated, but if it's a few minutes, we can get over a couple of minutes.
It's about, yeah, probably 3 minutes.
Okay, yeah, that should be fine.
Yeah, okay. So, okay, so now if you want to analyze a time lapse, experiment. If you want to follow, for example, cell growth, you can do the same analysis on the next time point. So here I was using the T7 time point and the T8 time point. Just make sure that you open in the right window. So your first time point has to be in mesh 1, so in this space, and the second one will be in stack 2, so mesh 2. So here I did this segmentation for the second time point only on the first cell layer, and as in 2D, you just make sure that the second time point is below the first one. And what is changing here? So there is no change about the parenting. So in stack 2, you click on parent. That will remove the colors because you don't need the label color of the cells. You just need the label of the first time point that you will match with the second one. You play also with the opacity of your sample. That will help you to visualize through your sample to find the same cell shape in your 2 time points. So here I make sure that I find the right one in this sample. That was pretty easy to find them, and you can just grab the label of the first time point into the second one. So if I accelerate it a bit, at the end you will have a match between the 2 time points. So for example, this cell is this one, the yellow one is this one. You can also, if it is helping you, change the scale of your sample. If, for example, you had too much growth in your second time point, you can just click on scale on your stack 1 or stack 2, depending on which one you want to modify. It does not affect anything. It's just a representation of your sample. Just make sure when you save that you are not saving with the scale, otherwise that will change and affect your results. But here you can change to have a perfect match of cell shape. That will help you to find the cell in your 2 time points. So then you can do the heat map classic also. You are interested on the volume changes, but what is different from before is that you're following the change of this volume. So click on change here. We are working on stack 1, so I just selected increasing here. I does not change anything in this area, and then click on OK. So it's giving this information. Then I do so the inverse. I work in the stack 2, but then I compare the stack 2 to the stack 1. So I select the decrease information here. So I have the same information in both stack. That is totally normal. And here, because I probably have a mistake about the red cell here, I just changed the heat map set range to clearly visualize the cell growth in the 2 time point. I does not have time to show you how to correct the problem with this red cell today, but please make sure that the result you have is relevant and it's logic also by checking your 3D segmentation with your PM marker and make sure that you don't have any problem. For example, with the stomata, there is a lot of easy problem to have. So sometimes you can see that there is a lot of cell growth in this kind of cells, but make sure that it's real and not just a problem from the analysis. Yeah, so as I said, I just placed here the different tools that we are using when we are doing the analysis, when we move the sample, for example, when we select the cells, when we change the label of them, or the tool that we are using when we do the parenting. So thank you, everyone. I hope that it's going to help you for your image analysis in 2.5 and 3D. So if you have any
Thank you very much. Yes.
Thank you.
We have answered a lot of questions already in the live chat, so I don't know if there's more questions. If you can type them or ask them here. If not, you can always contact us later if you have some more specialized questions. Yeah, because this is just we just scratched the surface here. Yes.
Yeah, and it can be also a lot of information in like a short period of time. So you will probably try at home to do this analysis and have problems, of course. And I hope that you have problem. Otherwise, you will not learn how to do the segmentation. And if you have any question at that time, you can just contact us and we can help for this.
Awesome. Well, thank you, everyone. And this is a reminder, this was recorded and will be on the YouTube and the PCA website. We will send the links with all the information to all the participants once it becomes public and it's live online. So yeah, thank you all the panelists. This was wonderful. It was really helpful. Gonna get an initial view into the morphogenics and kind of how it actually works and how cool and powerful program it actually is. And yeah, that's all we have for today. And then hopefully we are shooting for September to have the third workshop focusing on the use of the AI in image analysis. Okay,
Thank you very much.
Thank
Yeah,
you.
again for your time.
Thank you.
Bye.